Welcome to Developmental Biology. Today, we are going to explore a fascinating and crucial medical procedure known as amniocentesis. Imagine being able to safely peek inside the womb to check on a developing baby's health long before they are even born. This procedure does exactly that, acting as a temporary window into the hidden world of fetal development. We will uncover the biology behind how it works, why it is so important for expecting parents, and even dive into the mathematics used in the laboratory to analyze the results. Let us get started. To understand how this works, let us start with a simple analogy. Imagine you have a sealed, opaque fish tank. You desperately want to know if the fish inside is healthy, but you cannot open the tank without causing distress. What do? Instead of disturbing the fish directly, you carefully extract a tiny sample of the water it swims in. Because the fish naturally sheds scales and waste into that surrounding water, analyzing the water tells you almost everything you need to know about its health. In developmental biology, we use this exact same brilliant concept to check on a developing baby without disturbing it. Now that we understand the basic concept through our fish tank analogy, let us look at the actual medical procedure. Amniocentesis is a specialized prenatal diagnostic test. During pregnancy, the baby is surrounded by a protective, warm liquid called amniotic fluid, which cushions the fetus and helps its lungs and digestive system develop. Just like the fish in the tank, the baby naturally sloughs off cells from its skin, lungs, and digestive tract into this fluid. By using a long, thin needle guided by real-time ultrasound, a doctor carefully withdraws about 20 milliliters of this fluid. Scientists can then analyze the DNA within these fetal cells to assess the baby's genetic health. Before we move on to why this is so important, let us visualize the procedure with this diagram. Here, you can see a cross-section of the mother's abdomen. Notice the ultrasound transducer resting on the skin at the top. This is crucial because it provides a live video feed, ensuring the doctor knows exactly where the baby is. Inside the womb, you see the fetus, safely cushioned by the amniotic fluid. The placenta is located on the right. Observe the long amniocentesis needle entering from the left. Thanks to the ultrasound guidance, the needle carefully bypasses the fetus and the placenta, drawing up just a small sample of that cell-rich amniotic fluid for the laboratory. Having seen how the procedure is performed safely, we must ask why doctors recommend it. Before amniocentesis, there were very few reliable ways to know if a baby had a severe genetic condition before birth. Today, this procedure is incredibly important because it allows us to detect chromosomal abnormalities, such as Down syndrome, also known as trisomy 21, with nearly perfect accuracy. It also helps identify neural tube defects, like spina bifida, by measuring specific proteins floating in the fluid. Knowing this information early is life-changing. It gives parents and medical teams crucial time to prepare for a child with special needs, plan for immediate medical interventions right after birth, or make deeply personal reproductive decisions. Now that we know what conditions we are looking for, let us step into the laboratory. When the amniotic fluid is first extracted, there is a practical problem. The number of fetal cells present is usually too small to immediately analyze under a microscope. To perform a karyotype, which is a visual picture of the chromosomes, scientists need a large, healthy population of cells. Therefore, they must grow or culture these extracted cells in a laboratory incubator. In biology, the growth of these cells is not linear. It follows a standard exponential growth model. This means the cells multiply rapidly over time, and we can use mathematics to predict exactly how long we must wait before testing. This biological reality of cell multiplication leads us directly to our first mathematical formula. The exponential growth equation allows us to model how the cell population increases. In this formula, n of t represents the final number of fetal cells available after a specific amount of time has passed. And 0 stands for the initial number of viable fetal cells successfully extracted from the sample. The letter e is a mathematical constant, the base of the natural logarithm, which perfectly describes continuous growth processes in nature. The variable r represents the specific daily growth rate of the cells in the incubator, and t is the time elapsed in days. If the initial cell count is higher, or if we wait for a longer time, the final number of cells will increase exponentially. Before we look at the deeper algebra, let us see this formula in action with a concrete example. Imagine a laboratory receives a sample with an estimated initial count of 500 viable fetal cells. To successfully perform a clear karyotype for Down syndrome, they require a minimum of 10,000 cells. The specific growth rate in their incubator is 0.3 per day. We want to find out how many days they must wait. First, we substitute our known values into the equation. 10,000 equals 500 times e to the power of 0.3t. Next, we divide both sides by 500 to isolate the exponential term. This simplifies the left side to 20. Physically, this means the cell population must grow to exactly 20 times its original size before the genetic analysis can begin. Now that we have simplified our example equation, let us understand the mathematical reasoning required to solve for the time variable, t. Right now, t is trapped up in the exponent. To bring it down, we must apply a logarithmic function. We take the natural logarithm of both sides. Because the natural logarithm and the base e are inverse functions, they effectively cancel each other out on the right side of the equation. This brilliant mathematical property leaves just the exponent, 0.3t, on the right, while the left side becomes the natural log of 20. Finally, we divide the natural log of 20 by the growth rate of 0.3. The calculation gives us approximately 10 days. This tells the anxious parents exactly how long they must wait for the results. So we have proven how long cell culturing takes, but cells are not the only thing we analyze. Amniocentesis also checks the fluid itself for specific chemicals produced by the baby. One of the most important markers is alpha-fetoprotein, or AFP. If the levels of AFP in the amniotic fluid are abnormally high, it can be a strong indicator of a neural tube defect, such as spina bifida. Because the volume of fluid extracted can vary slightly from patient to patient, doctors cannot just look at the total mass of the protein. Instead, the laboratory must calculate the exact concentration of AFP in the sample to compare it against standard medical safety ranges. This requirement to standardize our measurements brings us to our second formula. To find the concentration of any substance dissolved in a fluid, we use the basic concentration formula from chemistry. Conceptually, it simply divides the total amount of the substance by the total space it occupies. In this equation, C represents the final concentration that we are trying to determine. The variable m represents the total mass of the specific protein, which in our case is measured in micrograms. Finally, the variable v represents the total volume of the amniotic fluid sample that was extracted, measured in milliliters. If the mass of the protein increases while the volume stays the same, the concentration goes up, which could signal a medical issue. Now that we have our concentration formula, let us apply it to a clinical example. Suppose a doctor extracts a volume of 15 milliliters of amniotic fluid. The laboratory measures the total mass of AFP in this sample to be 300 micrograms. We know the normal safe range is between 10 and 15 micrograms per milliliter. To see if our sample is safe, we substitute our known mass of 300 and our known volume of 15 into the equation. Dividing 300 by 15 yields a concentration of 20 micrograms per milliliter. Because this calculated value of 20 is strictly higher than the normal upper limit of 15, the doctor would flag this result as abnormal, indicating a higher risk for a neural tube defect. We have covered a lot of ground today, so let us review our key takeaways. First, we learned that amniocentesis is a powerful diagnostic tool that safely samples amniotic fluid, acting as a window into the womb to assess fetal health. Second, we saw how it is crucial for detecting severe genetic conditions and neural tube defects long before birth. Third, we explored the mathematics behind the medicine, using exponential growth models to predict how long we must wait for fetal cells to culture. Finally, we used concentration formulas to standardize the analysis of chemical markers like AFP. By combining biological principles with quantitative analysis, modern science provides profound insights that safeguard early human development. Great job following along.